again. Welcome to another episode of Gung Ho. It's April 10, 2017. I have a very special guest, Linda Frank. She is an expert in forensic science. She's an author. She's a writer, and we're becoming good friends. All right, Linda.
Hi, Joe. It's good to be on the show. Thank you very much for having me. It's a very interesting topic we're going to talk about today.
I just want to ask you a few questions about your pedigree. Where were you born?
I was born in Queens, Long Island.
Wow.
I'm a Long Island girl. Never left.
And what high school did you graduate from?
I went to a girls' Catholic high school, Mary Louis Academy in Jamaica.
I know where that is.
Yeah.
And college?
St. John's.
And I know now that you taught.
Yes, I taught in public school. I taught in Seaford schools, which is, Seaford is right between Massapequa and Wantua, South Shore.
I know.
And I was there for 30 years. I started teaching forensic science there as an elective, and I was so interested in it that I took a number of courses, and then I proposed a course after I retired from the public school system to Hofstra. So I taught it at Hofstra for 11 years, and I also taught a course in biotechnology, which is DNA technology. So I really got to understand and learn about and investigate DNA technology.
DNA. When did it appear for the first time?
All right, DNA actually was discovered in 1898 by a scientist by the name of Michener, but he really didn't know what it was. And it kind of just sat there for a while, and I suppose research scientists were interested in it because it came from the nucleus. And our bodies are made of cells. We have trillions of cells, but the control center of the cell is the nucleus. It seems that all the activities of the cells center around that nucleus. So they got the DNA from the nucleus, so they kind of thought this must be important. So they researched it, and by 1958, a team of scientists, Watson and Crick, who almost everybody knows those names, they won the Nobel Prize for actually figuring out the structure of this DNA molecule, which is a very large molecule. And they won the Nobel Prize based on photographs taken by a female scientist by the name of Rosalind Franklin. She used a technique called X-ray crystallography, which is really, in simple terms, it's just taking pictures of molecules.
I got one question before we go further. This molecule, is that part of our genes?
Yes. Well, this is the way it works. You have a cell. The entire body is made up of cells. The cells are what organize into tissues, into organs, and that's how our body functions. All living things are made of cells. The cell has a central part called the nucleus. Inside the nucleus, when the cells are dividing, because they have to divide to keep you alive, you have to be making new cells all the time. We see structures called chromosomes. Everybody knows that term, chromosome. We talk about chromosomes all the time. And you, as a victim of Agent Orange, know that you have probably suffered some chromosomal damage. The chromosome is actually a structure where the DNA is organized. And the chromosome is made up of smaller units called genes, and the genes are made up of DNA. So the functional part of the chromosome is really the DNA. I have a really nice picture of that in that PowerPoint. Maybe they can cue that up while we're talking. So that's really how it works. Everything that happens in a cell happens because the DNA enables that to happen. If you look up at that picture there, what you see is a cutaway section of a cell on the left. And in the middle is that sort of pinky purple round structure. That's the nucleus. And inside the nucleus, if you go to the diagram on the right, the X structure, that purple X, that's a chromosome. And if you unravel the chromosome, you end up with a DNA molecule. And the very right side of that picture shows you a portion of the DNA molecule that has two yellow strands. It's kind of like a ladder, and then it has rungs, or it has vertical bars that go down. And the code is on those vertical bars. If you took a DNA molecule and you pulled it and stretched it, think about a slinky. When you were a kid, if you had a slinky, the most tempting thing to do with the slinky was to pull it apart. So think of the slinky as a DNA molecule, and as you pull it apart, you get kind of a ladder. Okay, so DNA is like that. If you pull it straight, its normal structure is like a spiral staircase, and then if you pull it apart, it's like a ladder. And the uprights remain the same all the time, but the rungs change, and the code is in the rungs of the ladder.
You mean the code for each person is different?
Yeah, well, this is the trick, okay? Your gene for insulin, for instance, is the same as my gene for insulin, because I don't think either one of us has diabetes, right?
No, I
Okay,
don't.
so we have a normal gene for insulin. The genes, and what a gene does, a gene has one specific purpose, and that purpose is to make a protein. The instructions from the DNA molecule are actually sent out to another part of the cell. Inside the nucleus, what the DNA does is it just doesn't sit there. The chromosomes will unravel, and when they do that, that's called the resting stage of the cell. This is a misinterpretation of what's going on in the cell, because when the DNA unravels, it duplicates itself from the raw materials from food that you eat. There's raw materials in the nucleus, and the DNA duplicates itself. That's so that the cell can divide and the new cells will have the right kind and number of DNA. At the same time, it's making a messenger molecule. This is all amazing to me, that all this stuff is going on while we're sitting here, and hopefully it's going on correctly. So this messenger molecule then goes to another part of the cell called a ribosome, which is a term, just if you think about it as a protein factory. So the instructions from the DNA are really instructions from a gene to make a protein. And what's a protein? We're made of proteins, the structural material of our body. If you're a bodybuilder, you drink protein shakes. Why? Because your structural material is made of proteins. And also, all the chemical reactions in the body are controlled by proteins called enzymes. If you're lactose intolerant, when you take that lactate pill, that's an enzyme. Enzymes control all the chemical reactions in your body. And so it's important that that gene has the right instructions to make that enzyme, and also hormones. So those are the three proteins that really make up living things and control them. And that's why DNA is called the code of life, because it has the code, transfers the code to the protein factories. The protein factories make the proteins, and we function.
Okay, so this is incredible. So every cell we have has our DNA in it.
Yeah, well, except your red blood cells. Red blood cells, when they pop out of the bone marrow, they lose their nucleus. And the reason for that is because it increases the surface area of the red blood cell. Function of the red blood cell is only to carry oxygen. So the greater the surface area, the more oxygen it can carry. So this is like an evolutionary development. Red blood cells have a nucleus when they are made, but before they mature and leave the bone marrow, they lose the nucleus.
So do they have DNA after they're made?
They have DNA when they're made, but they lose it. That's why red blood cells die. Red blood cells only last for about 6 weeks, and they go to the liver, and the liver changes them into bile.
Do law enforcement use blood when they're looking for DNA matches?
Yes, but you see when they use the blood, there are other cells in the blood. The white blood cells have a nucleus, and that's the nuclear material that they get from the blood. But skin cells are better. Sperm cells are excellent for sources of DNA.
Saliva?
Yeah, well, that's called a buccal swab. When they have a suspect and they say, okay, open your mouth, and they swab the insides, because the lining cells on the inside of your mouth are always coming off. So that's a really good source of DNA.
Wow. All right. So when you were teaching forensic science, can you tell us, what is forensic science?
Forensic science is the science that's used to produce an argument for court. So in other words, when there's a case, it doesn't even have to be criminal, it could be a civil case. There's evidence, there's always evidence. So forensic science brings in all branches of science to process the evidence. If it's fingerprints, we're using photography. Okay, if it's ballistics, we're using physics. If it's explosives, we're using chemistry. And if it's blood, we're using serology or DNA. So all of those branches of science are brought in to deal with the different pieces of evidence so that the prosecution and the defense, because they have the ability to, as you well know, you're a defense lawyer, to produce this argument in court, because the word forensics means debate or argument.
I didn't know that.
Yeah.
That's what it,
Yeah.
wow.
Amazing, huh?
I know there are a lot of TV series now about forensic evidence.
Oh, yeah.
But in New York State, or in the city, they were called the crime scene unit.
Mm-hmm,
and
right.
they would respond to every murder and sometimes other crimes.
Mm-hmm.
And on occasion, the evidence that they gathered was helpful to me and undermined their prosecution.
Well, if they're doing their job correctly, okay, if they are following the protocols of the crime scene unit is supposed to go to a crime scene that's already been isolated. The first step is to isolate that crime scene, then record it. The crime scene unit should be recording it in as undisturbed condition as possible, because usually if there's a violent crime committed, the police come in and they pretty much disturb the crime scene. But if the technicians are doing their job correctly, and there is a protocol, every different type of evidence is packaged differently. Like, you don't put a bloody shirt in a plastic bag because you cut off the air and molds will grow in the blood and destroy the proteins and you can't process it. So they know what to do. And if they do it correctly and then it goes to the lab, and there's something called chain of custody, where every person who touches a piece of evidence has to date it and sign when they did it. If they do it honestly and correctly, what ends up in court, analyzed, should be what they picked up at the crime scene.
My experience is that all the experts that have testified truthfully. They just, this is the way it is, this is the way it falls.
Well, that's what science is.
Yeah.
Science should not start out with an opinion. Science should start out completely neutral, gather the evidence, and make a conclusion. But we're human.
We've got to talk about that.
Yeah,
All right, I
human,
want to know
right?
something about, this isn't just me, because just recently, a lieutenant that was with me in officer training school, OCS rather, basic school, then went to Vietnam, and on his second tour disappeared in Laos. They just found his remains, and they made a positive identification. How can they do that?
Okay, well that's on the PowerPoint, so that they could really, in the control room, cue up the PowerPoint, we'll talk about it. There is a special division of the Armed Services called the Armed Forces DNA Identification Lab, okay? And if you look up there, what they do is they will use 3 branches of science to identify remains. Fortunately, in 1988, the governments of Cambodia and Laos and Vietnam allowed the Americans to come in, and they knew where the different crash sites were. The military knew that as well, and they knew where the mass graves were, and they were allowed to go in and investigate those. They use archaeology. Archaeology investigates not only bones, but personal effects. For instance, if they can find personal effects in that grave, along with the service person, then that can tell them what the identity was. So they look for dog tags, they look for wallets, they look for personal effects of different kinds, and that's archaeology.
Look for
Yeah, your dog tag. Yeah.
dog tags.
Right, exactly. And sometimes the enemy will take those off the body because it's just a retribution. It's not a nice thing to do. That too. And also weapons can indicate who the person is, and also the armed services, they have records of the people who went into, certain operations and why. So that's archaeology. Then they go into anthropology, and anthropology is a study of the bones. Okay, so they look at the bones to make sure that they're looking at in some cases a Caucasian person, not an Asian person. That doesn't work so much in the service anymore. But the Asian person could be a Vietnam person in there. So they look at the bones, And then they know that there's a slide in there, there's a picture, it's a funny picture of a skeleton. And if you go to that one, they know that certain bones will yield a better DNA than other bones. So then we go to the DNA lab. And what they do is they pull the DNA out of the bones. The bone marrow has DNA. And then they use a process where they're not really, see, there it is. But that, I mean, you can't really read it from here, but the different colored bones indicate the best sources of DNA. And usually it's the long bones, like the green femur. Femur will, give you some good DNA. What they really check is they check what's called non-coding DNA. We used to call it nonsense DNA, but now we know that the non-coding DNA that's associated with the genes. Every gene comes with like traffic signals around it, and that non-coding DNA occurs in different variations. Let's take gene X, okay? There's also a slide up there. It shows a gene A, B, and C, and then it has X's in front of it. The guys in the control, they'll find it. And so we find out that in the human population, those traffic signals for that gene may only occur 4 times, but the 4 times that it occurs represents a different percentage of the population. So if we take 20 genes and we make a mathematical calculation of the probability, which segment of the population that represents, see, there it is. What we're going to do is we're going to get a pretty good mathematical calculation of the reliability of that person's genetic identification.
Are you talking about race now?
No, it has
Male?
nothing to do with race. That these pieces of DNA that they study are really the result of medical research. They've studied them so much that they have statistical data on them. See, in order to do this, you have to have statistics from the population. You have to be able to say that this person's DNA represents 35% of the population in that location. Then you go to another piece of DNA and you say, okay, that person represents 10% of the population. And every time you come up with a fraction, you're multiplying the bottom number. So eventually you're getting a number that's like 1 out of 7.5 billion. And that's the human population.
Excuse me, they don't compare the DNA that they recovered from the bone marrow to any of his relatives?
Oh, yes, they do.
Oh, they do.
But that's the first step is to do the person's profile, the DNA profile of the skeletal material that they have. Then what they do is they will go to a relative. And now, suppose they have no idea who this person is. What happens is they have asked relatives of POWs and MIAs to contribute DNA material. And that DNA material can come from the mother, because there's a special way to track the mother's DNA and relate it to the son. They can, if they get a father, they can take the Y chromosome, which belongs only to males, and they can track the DNA back to that missing person. And then they can also take what's called autosomal DNA, which is just total DNA. So really they have 700,000 sites on just your body DNA that they check, and then they have the male DNA and the female DNA. If they are able to get from a mother, a father, a sibling, okay, and if they can't do that, if they can get, suppose they have baby hair from the missing person, mother's first haircut. They save the hair, they save the baby teeth, that's all DNA. Eyeglasses, if they have a pair of their eyeglasses, you're always putting them on, you're scraping skin cells off. If you lick an envelope, if you wrote letters to your relatives when you were in Nam, right? You lick the envelope, you lick the stamp, you sent it home, your DNA is there. So that's the comparison, the relatives' DNA. So they take the DNA from the bones, the remains, and they compare it. And again...
And how accurate is this?
Your DNA experts will tell you it's 99.9% accurate. because they're checking so much of your DNA. And this is the same thing, that same process that they use on Ancestry.com. You know, when they do those commercials on 23andMe, Ancestry, same technique, exactly the same.
This is amazing.
So if you have a missing person, you have somebody you know who's a POW or an MIA, what you can do is you can contact the Air Force DNA identification lab, and you can ask them to send you a kit, just like Ancestry.com does, and you can contribute the DNA to them, and they can put it in their database, so that if they have a person, they have no idea who it is, then they run it through this, you know, the computer runs it through the database, and they can
This is ongoing all the time.
Yeah, well, if you look at if they come up with the second slide actually gives us 81% of the DNA they have tested, that they have found, the remains that they have found have been identified. In the Korean War, it's 89%. In the Cold War, I can't remember the figure. If they come up with it, I'll know. But in World War II, only 4%. And the reason why is so many people were lost at sea. It's the next one. Keep going. There it is. You can't really read the numbers, but I think it's, Cold War is like 94% or something like that. It's a high number. But World War II is a low number for two reasons. One is how many people were lost at sea, and also how many people were buried or lost or will never be recovered. Those remains will never be recovered because it was so long ago, 70 years ago, 75 years ago.
Okay.
So this is a wonderful thing that they're doing, because they're giving people closure.
I want to switch now to how DNA is being used in the courtroom. Start out with the Innocence Project.
Mm-hmm.
Could you tell us what that is?
Well, the Innocence Project was started by, not Dershowitz, the other one. What's his name? Barry Schechter. Barry Schechter.
Yeah, Barry.
That's our audience. Okay, and he was a lawyer at the OJ trial, and what he did is he had his law students looking into cases where men had been convicted and incarcerated. They were in jail for either a rape or a murder, a violent crime, and they maintained their innocence. They said, we are innocent. They would never get off that. Sometimes when they're convicted, they just say, okay, I did it. And so because they saved the evidence for so long in all of these crimes, they went to the evidence and they had the evidence DNA tested. And in some cases, it showed, yes, they were evidence. The DNA of the sperm or the blood, the evidentiary evidence, did not match the person who was in jail. So over 100 people have gotten off on this.
And, of course, if it doesn't match, that's like a definite thing. You
Absolutely.
got the wrong person.
I believe that what the courts do is, if it's not a match, it's not a match, okay? But if it is a match, it has to have corroborating evidence. Courts are very cautious in accepting evidence, accepting techniques of analysis. And the case that you were telling me about, the evidence was tampered with. It's similar to what was claimed in the OJ case, where in the OJ case, the defense claimed that the detectives took blood from the crime scene and took it to OJ's house and sprinkled the blood on OJ's socks to incriminate him.
I think they did something worse than that. They think they took it from the crime lab. Oh.
That story I hadn't heard, but I had heard the one
I'm
about...
not sure.
Yeah.
But I want
so
you
it
to...
can be tampered with.
I want you to explain, like, how could that be? How could, you know, they recover, say, what are they? Well, in that case, what happened is they asked the defendant to give a blood sample. And then they said that his blood matched some sperm that was found on the victim's garments. And he maintained his innocence. But I found out, it was not part of the case, that these detectives really had it in for him, and they went to his home with a search warrant to gather evidence for a crime of keeping a rental vehicle beyond the term of the lease, which is like stupid. And
Yeah.
They were in there the whole day, and they found nothing. Now, I knew that. The jury didn't know that. Now, and this kid, you know, call him a kid, he was a very young man, smart, you know, sort of an attitude. but he was really sexually active. If you go into his bedroom, you're going to find a lot of sperm, you know.
Probably.
And then what I found out, I had an expert testify. He told me about a thing called mapping, where you could say there's a stain there, right? Mm-hmm. All you have to do is take a cloth.
That's right.
Touch it, and then put it over here, and then all of a sudden, I'm the suspect.
That's one of the greatest flaws of DNA evidence, is it's so easy to contaminate it. I mean, even if I accidentally touch, you know, I'm scratching my head, I get my skin cells on my fingers and I accidentally touch a piece of evidence, my DNA is transferred there. So how this happens is because someone gets access to the evidence locker and is able to go in and tamper with the evidence. In other words, take some of the evidence out and transfer it. You know, people are human, and a lot of people are not nice, and so things like that happen. Bad things happen.
So this young man got 20 to 40 years.
Oh,
I
Lord.
believe he's innocent. And the only evidence against him really was the DNA evidence, which people don't realize is circumstantial evidence. But circumstantial evidence can be sometimes stronger than direct evidence. Mm-hmm, mm-hmm. That's all they had. Yeah. And he got convicted for 2 rapes. What?
You have 1 minute.
Oh.
On the clock.
All right. All right.
Yeah, so, did I answer all your questions?
You know something? For the first time, I finally understand what DNA evidence is.
Well, I'm really
where
glad.
It is.
I'm going to send you that PowerPoint. I'll email it to you because it has a lot of information about Vietnam and how they actually did the project. I have a picture of a site where they're digging out the bones and whatnot. It's very interesting. And also, at the very end of that PowerPoint is a website that people can go to get information on how to contribute. If you can get that very last slide in, because we have like 9 seconds left, that would be helpful to the audience. Bring that up while the show ends, because we have...
Well, it was a pleasure.
Yes, thank you.
I'd like to have you back again.
Yeah, sure.
Talk more about forensic science.
There it is. So if people can read that on the screen, on their TV screen, that's helpful.
All right.
Yeah.
Thank you very much.
You do nice work. Champion of the people, right?
I saw a familiar face walking on the avenue, both surprised by the way